All right. Thank you, everyone. Sorry for waiting. So I'm Brian Schilder. I'm a PhD student at Imperial College London. Today I'm going to be presenting our lab's work on navigating the rare disease landscape, a comprehensive approach to identify gene therapy targets based on cell type phenotype associations. So why should you care about rare diseases? Well, despite the fact that each individual rare disease, it only affects a tiny fraction of people, rare diseases as a group actually affect up to 10% of the general population. And because of the heterogeneity across rare diseases, it makes them quite difficult to study one by one. But over time, we've come to understand that over 80% of rare diseases have a strong genetic component. This opens up new avenues for treating patients in the modern era of genomics. What does rare disease care actually look like in the clinic? Well, let's walk through a specific example by breaking it down into the key stages. First, when the child is born, they're assessed for any clinical abnormalities. In this case, the baby is small for their age, has low muscle tone, and is underdeveloped in their genitalia. And neither of the parents have a history of any of these symptoms, or as we'll call them, phenotypes. The doctor suspects that these phenotypes may be caused by some rare genetic disorder, but they don't know which one. So they order some tests, including a genetic panel to search for known mutations and perhaps a tissue biopsy to assess a particular tissue. Unfortunately, these tests come back inconclusive and the child remains undiagnosed. The diagnostic odyssey can last for years or even decades, with a 75% chance of never receiving a diagnosis. So just progress here. So the doctor tries to manage the symptoms as best they can, waits until other phenotypes start emerging over time. And as the child gets older, these include late onset puberty, morbid obesity, and mild to moderate learning disabilities. Finally, the doctor is able to put together a hypothesis and basically identifies that the rare genetic condition in question is Prader-Willi syndrome. Rare diseases like this can often be really difficult to diagnose, as patient-to-patient presentation of phenotypes is very variable, and some genetic conditions have only partial penetrance of the associated genetic variants. So the child is now in their late teens. What treatments are available to them? Well, the most commonly prescribed treatment for Prader-Willi is recombinant growth hormone, but it doesn't treat all the symptoms. It doesn't work for everyone, and in fact, it's far more effective when the child is much younger, meaning the optimal time window for treatment for this individual has already passed. Obviously, there's a lot of room for improvement with this typical pipeline, so where do we begin? First, doctors need a way of communicating their findings with other doctors and researchers. This is especially important as there may only be a handful of cases for a given rare disease in the world. And having a standardized language to describe the phenotypes can go a long way towards rapidly identifying patients with the same suspected disease. The Human Phenotype Ontology, or the HPO, was designed to do exactly that and contains over 17,000 phenotypes that are put into a structured ontology, all of which have their own unique IDs. It's structured so that it's hierarchical. The high-level phenotypes are terms like, at the very highest level, phenotypic abnormality, and they're very, very broad, all the way down the tree as you descend down the branches of the ontology, getting to terms like short stature, tall stature, or even more specific. So let's go back to the example from earlier, where the doctor noted low muscle tone. In the HPO, this would be rephrased as neonatal hypotonia. Similarly, underdeveloped genitalia will be refined to male hypogonadism. Great, so the HPO is a very useful tool for communicating phenotypes, but in addition, it contains gene annotations for over 10,000 of these phenotypes. These genes are also associated with particular phenotypes. These genes are associated with particular phenotypes via reported clinical cases of diseases in which those phenotypes exist in a given patient. And these have been manually curated by GenCC to assign scores that numerically categorize the current strength of evidence supporting each phenotype gene association. Okay, so what do we have so far? We have a standardized ontology of the phenotypes that make up each disease. We have a pretty extensive list of genes associated with each disease's phenotypes. But what are we missing? Well, for the most part, these diseases, we don't necessarily know how these genes are causing the phenotypes. And specifically, we don't know which tissues or cell types are actually causing the phenotypes as opposed to those secondarily affected by downstream consequences. This makes these diseases both very difficult to study and prevents us from accurately targeting the causal mechanism of the disease through treatments. Fortunately, we have a lot more tools at our disposal than we used to. This includes gene therapy, which can basically be broken down into two main categories: those that modify the expression of disease genes, and those that directly edit the DNA to fix the mutation or replace the disease genes. Things like introducing transgenes through gene replacement therapy, or using CRISPR to edit specific base pairs. In addition, there's been advances in our ability to target the treatment to specific cell types using things like cell type specific epitope markers so that we only affect the cell types of interest. But what does cell type specificity actually get you when it comes to treatment? Well, you can have more targeted assessment of the abnormal phenotypes in expected tissues and cell types. You can order more directly relevant and accurate tests that investigate those cell types if you're a clinician. And this would allow you to come to a diagnosis much more quickly. And ultimately, you can use this information to design therapies that are more effective at treating the phenotypes associated with the disease, and importantly, have far fewer off-target effects in cell types and tissues that you don't want to actually modify. So, to basically summarize this, to treat the disease, we want to treat the phenotypes. And in order to treat the phenotypes, we want to treat the cell types to have the most targeted treatment possible. To briefly summarize, the objectives that we set out to achieve in this study were as follows. We wanted to identify the cell types underlying rare disease phenotypes. And we also wanted to prioritize the cell type specific gene therapy targets that were most likely to be effective. So how do we do that? Well, as I mentioned, we had the gene list for over 10,000 phenotypes from the HPO. To get the cell type gene signatures, we use published single-cell RNA-seq data from human fetuses, and then computed how specific the expression of each gene was to each cell type on average. And this Descartes single-cell atlas is particularly important because it's sampled from every major tissue system in the body and includes 77 cell types, which is fairly comprehensive. Also being a developmental data set, it's going to be very relevant for a lot of rare diseases that are primarily developmental disorders. We then fed in both pieces of data into the tool that we had previously developed called expression-weighted cell type enrichment analysis, or UC, which basically asks the question, is this phenotype's gene list more specific to cell type A relative to all the other cell types? And then it repeats that 100,000 times per phenotype cell type test through bootstrapping to ensure robustness. From this, you get a cell type enrichment p-value and fold change for every combination of phenotype and cell type. And after multiple testing correction very stringently, we ended up with over 8,000 different significant phenotypes associated with different cell types. There we go. Okay, so on to some specific results. So looking at the results altogether, we wanted to check a couple of things first. So as you descend down the ontology to more specific phenotypes, there are fewer genes associated with those phenotypes. And this makes sense since the very broad phenotypes like phenotypic abnormality or abnormality of the nervous system encompasses many subphenotypes within that. So it's kind of an aggregate of all of those. This is also reflected in the fact that more specific phenotypes tend to be enriched for fewer cell types. This is exactly what we're hoping for because it means that we can pinpoint causal cell types by breaking down diseases into their constituent phenotypes. And the more precisely we can do that, the more we can understand the disease. At the same time, more specific phenotypes show much stronger enrichment for the few cell types that they're enriched for. This makes sense because at very broad phenotypes, abnormality in the immune system again, they're going to be an average of all the other cell types, and the subphenotypes are going to be more informative. We also checked that the phenotypes from, for example, the abnormality of the nervous system branch of the ontology were primarily enriched for nervous system cell types, and indeed they were very, very strongly through a series of hypergeometric tests. And this was also very true for the cardiovascular phenotypes, being strongly enriched for heart and vascular cell types, and for the immune system phenotypes being enriched for immune cell types. And this gave us confidence that our results are actually capturing valid phenotype cell type relationships, so we can start diving into more novel results. So great, we've accomplished what we originally set to do, which is characterize the cell types underlying each phenotype in the Human Phenotype Ontology. But we also wanted to have a more translational outcome, so we decided to take this study a bit further by systematically prioritizing cell type specific targets for gene therapy. But we still have over 8,000 significant results, so we developed a prioritization pipeline that systematically filters these results at each level, from diseases to phenotypes to cell types to genes. And at the highest level, we selected only the most severe diseases and phenotypes. In other words, ones that have very early childhood onset, can cause death, blindness, cancer, intellectual disability, and so on. We then filtered the level of cell types, focusing on those that are terminally differentiated, as existing gene therapy methodologies tend to work best for cells that are not going to be replaced in about a week. Finally, we filtered based on gene size to make sure that the gene being delivered could actually fit inside a viral vector, which is currently the most common form of gene therapy. But this is likely going to change in the near future because there are some clinical trials going on right now that rely on technologies like CRISPR and prime editing, which will obviate the need to have to insert an entire transgene. Instead, you can just edit at the base pair level. We wanted to see how many of the gene therapies that are already out there, either in the market or in clinical trials, that our pipeline actually identified the, quote, correct target and in the same disease. And of the 57 gene therapies that are out there, our pipeline predicted 65% of them with the correct target. And while it's not guaranteed that all these therapies are going to work in the end, they'll make it past trials, it does indicate that we're probably on the right track for many of our target predictions. And even for the trials that are currently ongoing, our results can be informative for which cell types are likely going to be impacted by the treatment and which other off-target cell types might be liable to cause unintended side effects. So something for people to look out for. Let's look at a specific example of one of our more novel proposed gene therapy targets. So Neisseria infections are the leading cause of bacterial meningitis. Recurrent Neisseria infections have been previously linked to the complement system, but there's over 45 genes in the complement system, and they're expressed in a variety of cell types. So it's really unclear which ones are actually causing this phenotype. Okay, thanks. Our analyses were able to pinpoint the multi-scale mechanism underlying the recurrent Neisseria infections from the disease to the phenotype. And so you're basically able to trace this from the level of disease down to phenotype to the genes, but contextualizing it within these particular causal cell types. And these were hepatoblasts and ALB positive cells, which are basically precursors to liver hepatocytes. Besides identifying gene therapy targets, what else can we do with the data? Well, this also opens up some avenues for personalized cell type specific medicine. And how would this work? Well, you can take an individual's list of phenotypes aligned to the Human Phenotype Ontology. if they have a suspected disease diagnosis, say Prader-Willi syndrome. You can include that as well, although it's not required for this pipeline. And finally, if the child has any confirmed deleterious mutations in certain genes, you can include that information. And you plug all of this into an algorithm, which we included in our package, which essentially aggregates all of the enrichment results from earlier, and then computes a gene-weighted cell type specific score and ranks the most likely causal cell types. And this is helpful because not every patient has every symptom associated with a given disease. In fact, many diseases, phenotypic presentation is highly variable, even when you have the same mutation. And in this case, the algorithm nominated adrenocortical cells and islet endocrine cells as the most likely causal cell types. So the physician may now want to investigate further by taking a tissue biopsy, or perhaps ordering a series of tests that measures the level of hormones produced by these cell types. Finally, we wanted our impact to go beyond just our initial publication and have this be useful for other people. And for this reason, we launched the Rare Disease Cell Typing Portal, where researchers, clinicians, and even patients can access these results in a user-friendly interface. This includes several apps where you can search by cell type to find the associated phenotypes. You can also search by phenotype and see which cell types are enriched. And finally, you can explore multi-scale disease mechanism networks, like in some of the examples that I showed earlier. So what have we accomplished at the end of the day? Well, we first identified causal cell type specific disease mechanisms at multiple biological scales. We then prioritized cell type specific gene therapy targets that have an improved chance of success. Next, we enabled cell type specific personalized medicine recommendations that put minimal burden on clinicians, which is important. And finally, we provided all of the results as a public resource, and all of these are highly reproducible through a set of novel R packages that we developed that are all open source, and these are called HBO Explorer and Multi-UC. Lastly, I'd like to thank all of you for attending today. And here's a QR code for the preprint if you want to read more, as well as another one with my contact info. And I'm happy to talk more afterwards and take questions now. Thanks.
Thank you very much, Brian. A great talk.
Thanks.
Are there any questions?
So great, thank you. Great, both on the content and on the presentation. Well, good job. My question is, in Orphanet, they have frequencies associated to phenotype for diseases, and I was thinking it could be interesting to use also this information either as input or to validate the different cell types, maybe?
Yeah, so that was included as part of our pipeline. I kind of breezed over it when it came to filtering. Because there were some phenotype frequency annotations, and that can be at different levels. So there's frequency annotations of how frequently is a gene, a disease gene associated with a particular phenotype. There's also frequencies of how frequently is the phenotype associated with a given disease. Yeah, so we filtered at both levels. The problem is the proportion of phenotypes and diseases that actually have those annotations is very, very small. It's like 1% of the entire HPO.
I thought yeah, maybe from the disease level, it's 20% of the disease. Yeah.
Probably, yes,
right,
at the disease level, it probably is higher, but actually connecting the phenotypes to the diseases, I think there's relatively few that have those annotations. But yeah, so we do at least include that there. And the idea is we want to filter out, if we're looking at rare diseases, and we want to improve our chances of actually helping some population that's already quite small, you would want to go for the phenotypes that are relatively common. And you could also, remove that filter and go for any phenotype that occurs within that disease.
Okay, thanks. I have other questions, but maybe...
Are there any other questions? Oh, sorry.
Hi, great talk. I do have a question. It might be silly, but you're using the gene expression from cell types,
Mm,
and that is in native state, so wild type,
yes.
normal. So the question is, and not always in these diseases you have a mutation, but it doesn't change the expression at all of the gene itself.
hmm
Do you plan in the future, or is already included in some way effect of the mutation in the cell line in a pathological condition, so to speak?
Yeah, so that is something that we're missing here. We're really just using the fetal data set as a baseline signature of these different cell types, not necessarily what you would expect to see in the patient itself, but more as just as a way of identifying the causal mechanisms. But that piece of information that you're just talking about is important when it actually comes to testing things, because perhaps there's some markers for a given cell type that are ablated because of the disease. And you would want to know that information. So I think that comes down to designing tests that are actually aware of the more specific molecular mechanisms of that particular disease, whether it's a splicing mutation, it's knocking out the gene entirely, or some CNV alteration or structural variant. Like that's another level of information that we're not necessarily including here, but we could.
Any other question?
Thank you. I was wondering if you would benefit from contrasting the gene expression data that you have with the adult gene expression data and maybe identifying differentially expressed genes. Maybe this would be of even higher interest to you.
Absolutely. So part of the reason that we went with the Descartes data set was because at the time that we started the study, it was the only multi-system human single cell atlas. And in fact, it's a kind of a mix of single nucleus and single cell.
Yeah.
And single nucleus is not so great for identifying subtypes. So since then, there's actually been several adult and human fetal atlases that have come out, including Tabula Sapiens and the Human Cell Landscape out of a group in China that use microWellSeq. And that's actually whole cell. So we haven't actually run this pipeline using those data sets, but I have processed them, and I think it would actually be a really nice validation, because one of the strengths and limitations of the Descartes data set is that, as you saw earlier with some of the hepatoblasts and precursor cells, that's great if you're treating them prenatally. But usually by the time the child's born, those have all differentiated into hepatoblasts, or hepatocytes rather. So it kind of depends on what your use case is, but it would be, and it's not so much of a logical leap to say like, well, we don't have hepatoblasts, but we can test hepatocytes. But you would want to make sure that indeed the signature isn't so different from the adult version that it's not actually relevant. So yeah, we are planning on doing some of that as well.
Okay, thank you. And thank you for the great talk.
Yeah, thanks.
Any other question? Anais, if you want.
No, he asked my question.
Oh, okay. Okay.
So hi, and really nice talk. So normally with rare diseases, you also have this kind of scenarios. Maybe it's a bit outside of your talk, but where treatments are known to work, but you don't really know why in the molecular level, no?
Yeah.
So I'm thinking if the data you are presenting could be useful to try to analyze these kind of scenarios, and if you are actually doing this, maybe.
Absolutely. And I think this gets into the cell type specific personalized medicine.
Absolutely.
And not only in recommending certain treatments that are suspected to maybe act via a particular mechanism, but actually you could take some information about which treatments, well, rather, which phenotypes were responsive to which treatments and which were not across patients. Which is information that I don't have right now, but I imagine I could gather, and use that to inform, well, if this individual is primarily affected by this cell type, let's identify what these treatments are actually doing. And that's not even necessarily a rare disease problem. That's a medicine problem.
Absolutely.
We don't know the mechanisms of lots of commonly prescribed medications. And so, yeah, you could actually combine that information and, increase the chances of actually providing a treatment that's going to be effective. Because a lot of them right now, it's just trial and error. It's kind of based on population level. What works for the most people, that's our first line treatment. If that doesn't work, we try our next line treatment. And in the absence of information, it's just kind of a guessing game.